Lateral flow assays are low-cost, rapid, and easy-to-use tests which make them ideal for many different applications. In principle, lateral flow assays are immunoassays that utilize antibody-antigen interactions to detect specific targets or analytes. These targets can be anything from infectious diseases such as influenza or COVID to allergens or hormones such as HCG to detect pregnancy. The beauty of lateral flow devices is that they require no equipment. However, a reader may be required if a quantitative result is desired. An antibody is a protein produced by the body in response to the presence of foreign substances such as bacteria, viruses, or allergens. An antigen is the specific protein that is recognized by the specific antibody. This could be a protein on the surface of the bacteria or virus, or an allergen protein, to name a few examples. The most common type of lateral flow assays are sandwich immunoassays. In these assays, conjugate antibodies first bind an antigen. This complex then travels toward immobilized antibodies by capillary action. The immobilized antibodies bind the same antigen at a different binding site, thus creating a sandwich. The process of how this works in a lateral flow device will be explained in detail later. The main components of a lateral flow assay are the sample pad, conjugate pad, nitrocellulose membrane, and wicking pad. All these components are assembled together on an adhesive backing card, and strips are commonly assembled into plastic housing cassettes. Some lateral flow devices are placed directly into the sample in a dipstick format. Lateral flow assays are intended for many different sample types, ranging from clinical specimens such as blood, urine, stool, or respiratory specimens, to samples like food, water, or environmental samples. Make sure to read the package insert and intended use of the product you are using to know what specimen or sample type it is intended for. To run a lateral flow assay, first, the test sample is applied to the device via the sample pad. The sample pad may or may not be treated with certain agents to neutralize the pH of the sample and block non-specific binding interactions by molecules other than the target of interest. The sample flows through the device by capillary action. The sample will come into contact with the conjugate pad next. The conjugate pad contains a mobile antibody bound to a label. This labeled antibody is also referred to as the detection antibody. Once the sample comes in contact with the detection antibody, and if the antigen is present, the antigen will bind to the detection antibody at the specific site that the antibody recognizes. This binding site is also called the epitope. In most cases, the label on the detection antibody is colloidal gold. There are other types of labels as well, such as colored latex particles and cellulose nanobeads. The label is what allows the visual detection of the target or antigen. If the device uses a colloidal gold label, the color of the line for positives will be red. From here, the sample continues to flow through the device and makes contact with the nitrocellulose membrane next. The nitrocellulose membrane contains immobilized antibodies, striped and dried in a line format. The first immobilized antibody line is the test line. It contains the capture antibodies, which will recognize a different epitope from the same antigen recognized by the detection antibody. This is where the sandwich assay will occur if the analyte is present. As more and more of the antigen-antibody complex binds to the capture antibody, the color will start to appear and darken. Anything that does not bind to the capture antibody will continue to flow across the nitrocellulose membrane until it comes in contact with the second immobilized antibody line, the control antibody. Any detection antibodies that did not bind the antigen will be recognized by the control antibody and will be bound at the second line where a colored line will develop. The remainder of the sample will continue to flow through the device until it is absorbed by the wicking pad in order to prevent the sample from traveling backwards through the device, which can cause erroneous lines to form. Go to our website to see the lateral flow products Hardy offers for various applications and industry uses. Hardy Diagnostics is your complete microbiology supplier. Check out our product literature and catalogs in the links below. Subscribe to our YouTube channel for more tutorials and product videos. Remember to turn on notifications to be notified of new videos as they are released. Hardy Diagnostics is 100% employee-owned and has been serving microbiologists for over 40 years. To learn more about Hardy Diagnostics and the products we offer, visit us at hardydiagnostics.com.